Um, our next speaker is Professor Ravi Elangovan. Professor Ravi is a faculty in the Department of Biochemical Engineering and Biotechnology at IIT Delhi since 2010. He's a biophysicist by training, is actively engaged in developing methodologies and technologies for visualizing biological systems, and along with his team has developed multiple platform technologies for infectious disease diagnosis. With this, I'd welcome Professor Ravi to deliver his talk.
Good morning, everyone. It's really pleasure to come back to IIT Bombay in many ways, and it's almost feels like catching up with friends. So I have given some part of this talk earlier, a couple of years back. So I'm kind of updating the community again here after 2-3 years. So my experience of both from the innovation perspective, what happens in a lab, and where does it reach at the implementation stage is something I would like to give a small glimpse of. So let's start with the first slide. So this picture needs no explanation, right? So this is Gandhiji using microscope for leprosy identification. Microscopy, as most of the biologists here would know, that it is a fundamental tool. We use it to understand and visualize microscopes. It's very useful to get the cell shape and dimensions at the microscopic level. And though now we have a lot of molecular assays, we have get into it, but it is still, if you are using with the right kind of a dye, it can very highly specifically identify the particular pathogenic material. So, so this is one of the need for microscopy, and we had some good number of people related to TB, and this is a current status of tuberculosis diagnostics in India. So you have a 3-tier systems. You have primary, secondary, and tertiary care centers. And what is kind of important to notice is that our first line of diagnostics in tuberculosis is actually the microscopy centers. There are about 14,000 microscopy places where you actually identify a patient is tuberculosis or not. Now, we have seen diagnostics has got a lot of new meaning with the COVID, right? All of us actually got experience to all the tests, molecular assays, PCR. With TB, one of the thing is that we are the #1 in the world in patient load, and there is a lot of challenge in now treating them. So it is specifically in India, why we are actually having this high patient load is that just like COVID, if you are able to identify all the infected people and give them a treatment, you will be able to reduce the burden. TB is a very treatable disease, but every time we are actually looking out for this disease, the test that is currently used, the microscopy, is only about 60% sensitive. So 40% patients who are actually missed out in the system continue to infect, and we have a burden of 10 million populations being infected, right? So we need tests which can actually screen these patients effectively, and so this is our need statement. And I'm basically started my career with building this system called turf microscopy. So one of the things that we wanted to try out was, can we build it by ourselves? We can buy these ones for a commercial about a couple of crores, but we wanted to spend some time, learn how these things work when we build it ourselves. It took us a couple of years. This was 2012 when we learned how to build a microscope. So this gave us confidence that we can build stuff. So then we applied to a DBT project that we will try to miniaturize this turf microscope. And fortunately, they actually gave us grant and asked us to build. And this was done by a PhD student named Vikas Pandey. So we kind of miniaturized by assembling the optics onto the glass slide itself, okay? So turf is like where you have a continuous internal reflection of light. It's almost like your waveguide. So that's the optical fibers. You have the light continuously being internally reflected. So you can actually insert a laser light on the side of the glass slide and it propagates through the glass slides, okay? And so this is quite useful in microscopy because you need to excite the samples so that it can fluorescence. So what you saw, the previous version was actually the first version which we got it 3D printed. So it's a very important slide for all the innovators because the first time you actually come up with an idea and demonstrate it, it goes through a lot of changes. And I think this is our few prototypes that actually we have mentioned it here. But this is a thing that you should also allow, that when you have your first idea generated and you're establishing it, continue to improve on. And there are multiple things that you refine and it can actually take it to a more appropriate final system. So this is a TIRF system, compact TIRF system that you can actually place it on any microscope and you can actually generate a evanescent wave on the glass slide. So this is very useful for microscopy, fluorescence microscopy. And so this we wanted to apply it for TB diagnostics because the existing microscopy is done with a something called a bright field, which is all the scattered light. But if you use a probe called auramine and you can specifically label the tuberculosis cells and you can visualize these cells much more contrast. So that's where we had this particular prototype. Now, when we wanted to use this on clinical samples, I think Anidam was giving us this information that multiple stages you learn what could be the next challenge. So the sputum is a very thick sample. It is inhomogeneous. Everybody taking out a sample will actually get a different part. It can be a variability in the assay. So we developed this another solution for liquefaction of the sputum. So you can thin down, so you can release the bacteria that is trapped in the nets of DNA and other things. So we have now two systems. One is basically a compact total internal reflection excitation system. Another is a sputum thinning reagent. So this we call CLR. So we combine these two patents and we coined our product that's called CTB, so that you can visualize the TB effectively. And so we applied for patents. This is very important. This is whatever the technologies you build, file patent before you publish on. And surprisingly, in last couple of years, they have been granted also. Our patent system is working very fast these days. So this is how you compare it with the other existing systems. You have a bright field. This is called as a AFB staining, where you stain this basically, you can look for a pinkish cells, right? Now the other one is basically a commercial fluorescence microscope. You can actually use the same system and visualize the bacteria effectively. What we have done, we basically add up our adapter system to an existing microscope. We can upgrade this to a fluorescence system. And we have actually generated a lot of data to show this is basically the procedure and some of the performance data. We are actually able to visualize the cells as good as the existing fluorescence systems. And in some sense, we are also able to discriminate better the background because the TIRF waveguide is only penetrating about 100 nanometer in the surface. So again, this needs to be validated with the clinical specimen. So we compared it with the gold standards with the culture and gene expert, okay? And so we did a small sample, about 237, and we had about 76% against culture and 85% of sensitivity against a genes expert. Now, this is a kind of a good improvement with the existing system. The existing system with a bright field is about 60. You go to a fluorescence is about 70. And if you add the CLR and the CTB systems, we are hitting about 85%. It's still at not the most desirable part, but this is something very useful when we are actually looking at the scale of problem in India. So right now we are actually having many molecular assay tests that is being used for TB diagnostics. And if you have to detect about 10 million of TB patients, you need to actually test much larger than that. So you have to look at the health economics. And if we are actually looking at those many number of molecular assay tests, that the cost of burden is going to be very, very high. So that's where we still think that this microscopy-based stills have a utility. And if you compare the sensitivities at 85, we are working to improve it further. But the cost, turnaround time, and the existing infrastructure, all of it makes a convincing case. There is a good utility for the microscopy in the TB diagnostic space. So again, this is our story done till a couple of years back. And so we further wanted to improve how can we actually change this till system a little bit better. So microscopy is again still being used by a manual operator. And if you have to do 50 samples, you get in fatigue. There is a manual errors are all feasible. So the question we wanted to do is, can we digitize it? Can we automate it? And this was basically our next journey. So we looked at a lot of existing solutions that are trying to modernize the microscope. You have the system foldoscope. This is from a group from Stanford. And there are multiple variations of this one. So we have come up with this system, which is basically directly mounting an optical lens onto a CCD camera, which is readily available. And you can easily reach about 5 microns resolution. So the question next is that you need to have a fluorescence version of it. Now this is where again, the CTB we have actually added into this one. Existing systems are quite complex and bulky because that the optical collection itself is very large. So with our compact optical collection, we have also this perpendicular illumination. This is quite unique, and this is where that our profile is much, much compact. And so we added our turf into the system. So we are able to do the both bright field and fluorescence system. Now, this objective and the collection optics is actually almost a couple of centimeters. So that means that we could actually mount it on very simple motors that we ripped off a CD drive, and then we can actually automate the X and Y scanning. So you can actually scan the sample, X and Y, multiple positions. So this automation is also now, we have put it together. So again, the alpha version going to this kind of system, you have a box, and that you, along with the monitor, you can automate your completely microscopy. You can scan any number of images, and that images can be analyzed for TB cells. So this we have not yet started with the TB cells. We have just demonstrated for another large cell, again a critical condition. This is called mastitis in dairy sector. So in dairy sector, the animal can actually get an infection in the udder. If you continue on that, it can actually really hamper the health of the animal. So there is almost 30 to 40% of infection is there, and people use antibiotics rampantly in this dairy sector. So the challenge that we wanted to address is that when you have an infection, the neutrophils in the milk starts to rise, and you can actually count the number of neutrophils to basically see whether it is in a healthy state or it is actually going to mastitis infection. So we have a labeling protocol which involves a little bit of centrifuge, and then these are basically a milk sample, dairy neutrophil cells that is visualized using the same system. And this is not an AI system, it's just a normal cell identification and counting. We have done it with a manual and automated system, and we can actually precisely count the neutrophils in the milk, which can be used for identification of mastitis. So this we want to extend it further for the other cells in the future. And so there's some part of it work. And so this work, TB work was done with some of the collaborators, Professor Syed Asnain, Professor Nasreen Dakas. Vikas Pandey was the person who actually built the turf system. Now with the second team, we are actually building this mini scope where we are automating and digitizing it. So the journey continues. As we actually get up different valleys, we are hoping to jump one after another. Thank you all.
Yes. Thank you, Professor Ravi, for that very nice talk. We have time for a couple of questions. So if you have a question, please raise your hand and our volunteers will bring a mic to you.
So, nice talk, sir. So, I wanted to ask, when there is an infection, any infection, be it humans or animals, the neutrophils, they tend to shift to left or immature forms. So, using the system, currently, is it possible to know whether the neutrophils, they are mature ones or immature ones?
We have not done that, but I guess it's feasible by adding another second dye or some things like that. So we are right now using only a DNA-based label to identify the nucleus in the system. So there are people who used different nucleic acid labels to distinguish various types of WBCs.
I think there's a question on this side of the room.
Hello. Yeah. Hi, I'm Rupali from India Health Fund. It's a Tata Trust initiative. You've been in one of our one of our portfolio companies. We wanted to know that in One Health that you're trying to repurpose this entire innovation for use of in mastitis. So can the milk samples also be used for testing tuberculosis in bovine TB?
I'm sorry, I'm not familiar with bovine TB, but this is basically detecting cells that are present in the milk sample. So it's probably another new set of case that we have to examine. I'm not familiar about that.
Okay. So this is the same platform which is being used for now using milk samples to test mastitis.
Mastitis.
There's no new innovation in the platform as such.
So the platform is basically an automated microscopy system with the analysis system. So you don't have somebody to screen one by one and take a decision. So now by automating it, you reduce the time and also a little bit improve on the performance. So it's basically currently the microscopy is being all done manually and somebody is doing it. And that part we are trying to overcome by using this kind of an automated system.
Okay, that's very interesting. We'd love to learn more about
Sure,
the One Health as well, because I think we missed on that conversation. Okay, thank you so much.
All right, thank you. I think we'll take one final question.
Yeah, thank you, Professor Ravi. Actually, wish to know about the sensitivity and specificity of your system, microscope, which you have developed, comparative to sputum culture, which is gold standard, and gene expert.
Mm-hmm. You want to look at the data?
Yeah.
Okay. This is published in 2018, and go on if you have a specific question in mind.
No, actually, just wanted to know the comparative study of presently existing gold standard for the TB.
Okay. So
with
you have a culture
device.
test, which is basically, if you compare AFB, it's about 60%.
Yeah.
It's basically renowned and well published. So if you use a fluorescence-based test, it's about 70, because you are actually able to distinguish the cells more contrastly. Now, what we are doing is we are actually processing the sample with this CLR and using a new optical illumination system, which is helpful in actually reducing the background. We have a system which is going up to 85% in comparison to expert. Expert is now almost like a kind of a gold standard, or a molecular assay is being as a standard for identification of TB patients. So we are at 85%. That this is a part which is also a useful thing we should look at and evaluate, because the cost and time with the microscopy is in a very affordable system, and it could be a screening assay before could we take it to a molecular assay. So molecular assay is definitely there to stay with, because you need actually confirmation for resistance and other things. But a low cost screening can actually reduce the overall expenditure that we may have for the TB testing.
Thank you so much. Thank you.
Thank you. So now we will break for tea and coffee, which is on the ground floor foyer. Because the main focus of this event is discussions and interactions, we will not reduce the tea break time. So I request you all to please gather back here in another 15